Thanks, Drew. Thanks to ARM for allowing us to come in and give a presentation today, tell you a little bit more about our story. So I'm Tim Miller. I'm the President and CEO of Abeona Therapeutics, and we are a rare disease company. So like many other public companies here, I'll be making forward-looking statements. So this is our safe harbor statement, just to make sure that everyone's aware. So as a general corporate overview, Abeona is a leader in rare disease therapies. We have multiple clinical stage programs and a broad gene therapy pipeline. We have deep expertise as we look both in the U.S. and OUS for our regulatory programs going into Australia, Spain, and the United States. We do have a proprietary platform technology, the AIM, Next Generation AAV Viral Vectors. I'll touch on that just at the end of the conversation today. And we do have a very strategic focus for our rare diseases, very patient-focused as well. These offer smaller clinical trials and a shorter time to approval. Since in the past year and a half, we have brought in a number of pipeline products from both the metabolic, dermatology, and hematology programs. I'm going to focus today on the two programs that are highlighted with the red boxes for the treatment of MPS IIIA, or Sanfilippo syndrome, and epidermolysis bullosa. We wouldn't be able to do much of what we're trying to accomplish without the support of many of the foundations around the world. We are lucky enough to have been involved with these foundations and these parent groups from Spain to Australia, Mexico, Canada, Switzerland, and of course the United States. So to talk about our metabolic programs, as a gene therapy company, one of the things that we focus on is gene therapy is all about delivery. And it's about choosing the right virus or the right vector for the right disease, delivered in the right and appropriate manner for that particular disease. And so something that we like to point out is just that serotype, for example, and these AAV viruses do make a difference. Many of our diseases are central nervous system focused. So of course we're using the AAV9 virus, which you can see here for type 9. This slide shows a direct injection into the central nervous system, into the brain, and showing the tropism for AAV9 compared to some of these other viral serotypes to bind, inject, and be expressed in central nervous system. So again, we are using the AAV9 virus for our 3 or 4 lead programs. And you can see here that using this virus provides sustained CNS expression post-systemic injection. So again, with a lot of these therapies, you can give intramuscular injections, you can go through the intracerebroventricular route, intrathecal, or intravenous. Abeona's approach for many of our diseases is to use this as an intravenous approach to treat the whole body manifestations of the disease as well as the CNS, keeping in mind that AAV9 is the only AAV that is able to cross the blood-brain barrier. So for diseases with a profound central nervous system manifestation, you need to be able to use, again, a vector that's going to cross into the CNS and express in that tissue. So our 2 lead indications that are both clinical stage are MPS IIIA and MPS IIIB. These are rare lysosomal storage diseases that affect children. The lysosome is essentially the garbage factory of the cell. It's in charge of chewing up proteins and sugars and then turning them back out into the cell for metabolism. These kids are born with an inherited monogenic disorder. Sanfilippo is 4 different types, and again, we're focusing on type A and type B. 70% of these children do not reach the age 18. They have profound CNS and systemic manifestations, including liver and spleen enlargement. The kids are diagnosed between the ages of 2 and 6 and end up, as they progress through the disease, they have aggressive behaviors, seizures, loss of speech, vision, and inability to sleep. There are no cures currently available, no approved therapies. Incidence is about 1 in 70,000, and type A and type B are more common in North America and Europe. With our partnering hospital, Nationwide Children's Hospital, which has also spun out a few other AAV gene therapy companies that you may have heard of, have done a natural history study where we looked at 25 subjects and over a 1-year follow-up period. The study visits were at 0, 6 months, and 12 months and looked at a variety of neurocognitive, functional motor tests, spleen and liver MRI, enzyme activity, and of course looking at substrate levels. We use this as really our jumping off point for clinical trials. As you know, many these are small populations and with no treatment available, you're able to look at this, and this study helped validate our outcome measures for the clinical trials. The natural history study showed that all of these patients progressed over a 1-year period, except for one. Again, showing that you're trying to look within this window of the score of 30 to 80. This is part of our enrollment criteria for the clinical trials. But this, again, helped show that this is going to be a valid outcome measure for clinical assessments at 6 and 12 months. Similarly, looking at patient behavior assessments and the Vineland composite score, again, all of these patients declined over a 1-year period. So this is one of the, again, outcome measures that we'll be looking at in the clinical trials at 6 months, and we'll look forward to reporting that data in around second quarter next year. Importantly, the natural history study did show that in these children, an average of 100% increase in liver and spleen volumes over 1 year of follow-up. This is obviously significant. It's measured by MRI. So anything that can be done to show a stabilization of liver volume or even a reduction is certainly a clinically meaningful endpoint. And as an update on the phase 1/2 clinical trial, we have announced that we completed enrollment in the first three, enrolling the first 3 subjects at the low dose in cohort 1 at 5 e to the 12th VGs per kg. We have reported qualitatively and seen a reduction in urine and CSF GAGs, specifically the heparin sulfate. This has demonstrated that it did penetrate into the CNS, again, by seeing the reduction in CSF GAG. We've shown, and we'll report in the future, the quantitative numbers on how much reduction in liver volume and spleen volume, again, by MRI, and seeing increased in leukocyte SGSH enzyme activity levels. Coming up, as this cohort progresses, the drug has been well tolerated to date, and we are looking forward to, again, reporting on the evaluations of cognitive ability, adaptive functioning, motor and behavioral problems, and the pediatric quality of life. To talk about just 2 or 3 of our other metabolic programs, we also have a AAV gene therapy for Batten disease, or juvenile Batten disease. This is another lysosomal storage disease that results in the accumulation, again, of autofluorescent lipopigments. This disease initially presents as blindness. Also has very similar manifestations to Sanfilippo syndrome, behavioral issues, sleep disturbances, also cognitive loss, and premature death. This is just a slide from some of the preclinical data that recently was published, showing using an AAV gene therapy treatment very similar to the Sanfilippo programs, where these animals had improved or corrected muscle function, as well as additional data in this manuscript that shows that the survival deficit has also been corrected. We also recently announced the in-licensing of another program from the University of North Carolina, looking at infantile Batten disease. So Abeona is really building out our pipelines as both in the metabolic space with 2 programs in Sanfilippo syndromes and 2 programs in Batten disease. This is in the CLN1 version. So again, another lysosomal storage disease showing very similar virus and very similar efficacy, where these animals that were treated with the gene therapy had a normalization of survival over an 18 to 20-month period. And these studies are ongoing and will soon be published. So to talk now about our dermatology programs, we announced the in-licensing of a program for epidermolysis bullosa, also known as butterfly skin syndrome, which is a rare, devastating skin disease. These kids end up with blisters over most of their whole body, where their skin basically detaches and falls off. They have wounds that have sometimes not healed for 5 years or more. And with a significant amount of reduction or a significant amount of scarring. There are no FDA-approved treatments. So, any reduction in disease symptoms, such as improvements in wound healing, are considered clinically meaningful. So this particular EB program, EB101, is a clinical stage. We recently announced the treatment of the first patient in the phase 2 component. It is an ex vivo correction using a retrovirus to correct and insert the COL7A1 gene. So these cells are essentially, it's a sheet that the cells are pulled out by punch biopsy. grown, expanded, corrected, and then grown in and put onto a scaffold. This scaffold is then transplanted onto over the wounds. This is a little bit of the data that has been recently published, showing at baseline 3 months, 6 months, and 12 months. Each of these kids receives 6 grafts, and you can see that these grafts have lasted up to 12 months. So again, on the baseline, hard to use that pointer. Looking at the forearm, these kids, this is a skin that has been completely eroded, cleaned, and then the graft is grafted on top of that. And again, the graft has lasted now for over a year. As an update, in this clinical trial for RDEB, we have 5 adults enrolled with COL7A1 mutations. The primary endpoints consist of C7 and restoration of anchoring, and the absence of systemic virus at 3, 6, and 12 months. So far, all 24 grafts have been well tolerated by the subjects with no SAEs. At 3 months, 87% of the grafts showed significant wound healing, as defined by 70% healing compared with baseline, and at 6 months, 67% continued that healing. ABEO 101 does induce and sustains the C7 levels at follow-up, upwards of 60 to 80% increase in the actual protein expression in the transplant. You can see here where the lines are green, the restoration of COL7A1 production in these grafts as measured through a punch biopsy. And we also recently announced the acquisition of our next generation AAV platform, the AIM vector platform. This is a vector library of over 60 capsids, some of which have broad targeting to the CNS. It's a designer system that's used to direct evolution of natural AAV vectors. Some advantages of this are that we see increased gene delivery and efficient delivery. We do see target-specific targeting for some of the vectors, with the potential to reduce antibody response against them, as these are not natural. And we do see that these are, in some cases, superior to ones that are currently being used in other trials. So with that, I'd like to wrap up, and here's contact information in case anyone needs it. Thank you.